All right, hello, I'm Michael Schmidt, Senior Biotech Analyst with Guggenheim Securities, and it is my great pleasure to welcome Sam Kulkarni, CEO of CRISPR Therapeutics, to this year's virtual Guggenheim Oncology Day. Welcome, Sam, and thanks so much for joining us.
Thank you for having us.
So I'll start out with a broad question before we dive into the pipeline. And my first question really is, the Nobel Prize for Chemistry was obviously recently awarded for your co-founder, Emmanuelle Charpentier, for her work on CRISPR-Cas9. Could you, on the backdrop of that, could you talk about how your pipeline strategy has evolved, maybe relative to that of your gene editing peers?
Yeah, happy to. 2020 was quite the remarkable year for us as CRISPR Therapeutics, but also for the entire field. Just 10 years after Dr. Emmanuelle Charpentier elucidated the CRISPR-Cas9 technology together with her co-founder or co-inventor, Jennifer Doudna, this technology and the invention was awarded the Nobel Prize, which speaks to the transformative effect it can have both in biology and drug discovery, but in drug development and medicine. Additionally, beyond the Nobel Prize, we also had a data release last year with our hemoglobinopathies program, where we showed that 10 of 10 patients, 7 thalassemia patients and 3 sickle cell patients who were treated with CTX001 were effectively cured of their diseases. They had no symptoms after the administration of CTX001. And that, I think, when we look back at this time, will be hailed as a very important time in the history of drug development and medicine, is what I believe. So it's been a remarkable year. But if I just rewind the clock a little bit to your question around the evolution of the different companies using CRISPR-Cas9, you've had more than one company, and I think some of the decisions we made early have been paying off for us. Some of these decisions are to start with an ex vivo gene therapy, ex vivo approach, where basically we take the cells out of the patient's body, edit the cells, and put it back into the patient as a therapeutic. This is less risky from a technical standpoint than in vivo therapies because you don't have a delivery challenge. And the choice to develop our portfolio with an ex vivo program in sickle cell and thalassemia to de-risk the platform has given us a tremendous leg up. And then that also led us to immuno-oncology because we learned how to manipulate CD34 cells. And with that came the ability to manipulate T cells, where we harness the immune system to fight cancers. And in this approach, we do allogeneic CAR Ts, as you know. But one of the other important decisions we made was to keep it all in-house versus partner it. And I think that has given us tremendous value in terms of our portfolio and positioned us at the cutting edge of immuno-oncology cell therapy even though it's a crowded field. And lastly, that actually, again, has parlayed into a regenerative medicine portfolio, where we do many more edits to cells ex vivo. So all in all, I think these were some of the decisions that led to our portfolio as is, which is, again, different from the journey that some of our, I wouldn't even say competitors, there are peer companies in CRISPR using CRISPR-Cas9 have taken, but these choices have placed us, in addition to the execution around these choices, at the forefront of the technology and the potential of it.
Very good. Thanks for that introduction. So let's maybe start out with talking about CTX001, which is your therapy for rare genetic hemoglobinopathies, and that's partnered with Vertex 50/50 as well. So we've been following Bluebird Bio for some time. Maybe for those that are less familiar with the details, could you remind us what the key mechanistical differences are of CTX001 versus Beti-cel or Zynteglo, and whether those differences might potentially translate into a different clinical profile?
Yeah, thank you. I think we have 2 programs that have shown really good data, which is the Bluebird data in thalassemia and sickle cell, and our data, which we believe could be could represent best in class for these patients. To those who are uninitiated to the programs, the way I would characterize it is a scalpel approach versus a shotgun approach. In the Bluebird approach, they utilize a virus, a retrovirus that integrates into the genome, which carries with it an artificial form of hemoglobin. Recall that beta thalassemia and sickle cell are diseases of the beta globin gene, where in the case of thalassemia, the beta globin gene is deficient, and in the case of sickle cell, the beta globin gene is defective. And in the Bluebird approach, they overcome that by using a virus to carry a artificial hemoglobin, adult hemoglobin gene back into the genome. Now that is scattershot approach, whereas with CRISPR, what we do is make a precise cut in the genome where we, in a regulatory element of the genome, where we elevate fetal hemoglobin to overcome the deficiency or defectiveness of beta globin. And we do that by editing a region called the enhancer of BCL11A, which is only active in erythroid cells. I know it's quite technical, but essentially what it means is with our hematopoietic stem cells, we make an edit to something that's a regulatory element only in the fate of the cells when the fate of the cells have been decided to become erythroid cells. And in those cells, by making that edit, we elevate fetal hemoglobin. Now, how did we get this idea? The idea came from natural history studies. There were tens of families studied in various epidemiology studies around the world, where it was found that these patients and their families had the genetics of sickle cell thalassemia, but they were asymptomatic. And they were asymptomatic because they had a naturally occurring mutation that led to high fetal hemoglobin. So it's a proven approach that high fetal hemoglobin is safe and can overcome the issues with thalassemia and sickle cell. And these patients are, I shouldn't even call them patients, these people are normal, and they walk around without any symptoms. And so we were essentially recapitulating that phenomenon with our approach. And we believe that the data we show so far support our approach, and it has the potential to be best in class.
Very good. And in terms of safety, how should we think about the potential risk of off-target edits, which is something that's been talked about in the past, maybe compared to the random genome integration with lentiviral systems?
Yeah, we've done an incredible level of characterization around off-target. And for those who are not familiar with the question is, which is, we're using CRISPR, which is a scissor with a barcode, to make an edit in one part of the genome. The question is, does it only make the edit in that part of the genome, or does it make an edit anywhere else in the genome? Because we have 3 billion base pairs. The answer is we don't believe so. We've done extensive characterization across the entire genome, both in a biased and unbiased method, to see if there's any other off-target effects. And we haven't seen any in all our preclinical studies. And of course, we'll work closely with the regulators to ensure that that safety package is sufficient, both in the US and Europe, for that matter. And we don't see any off-target effect. So I would say that, compared to lentivirus, where every insertion is an off-target effect in a sense, with CRISPR, I think it's been very precise, and we haven't seen any adverse outcomes because of the off-target effects.
Very good. And I guess given that higher precision potentially, do you think regulators may be more open to maybe a smaller safety database, or maybe even shorter long-term follow-up than is typically required for viral-based gene therapies?
Yeah, I think the issue around our, from our trials of what the sample size is important. We're discussing that with the regulators now that we have this data set that is incredible, for lack of a better word. And the safety data set is for both thalassemia and sickle cell, right? The safety can be seen across both trials, whereas the efficacy population can be for each individual trial. And we think that from an efficacy standpoint, even a number as low as 20 in each study could be interesting. And across the two, you have a safety database of over 40 to 50 patients potentially. So I think that I think gives us quite a bit of confidence. In terms of duration of follow-up, we want to do the follow-up for a very long time. I think regardless of what the regulators say, because we just want to see what the effects might be of this therapy, but we don't anticipate any changes once the patient is treated and have made it past a year or so, but we would like to follow them for 15 years. And so that's something that we'll be doing regardless of what the regulators say.
Very good. And then on the efficacy portion, as you mentioned earlier, you did present some very impressive early data at ASH recently, and that was also published in the New England Journal of Medicine. I guess at this point, what are the remaining outstanding questions in either thalassemia and sickle cell, and how many of those might we be able to answer this year as we think about future updates from the studies?
Yeah, I think we'll continue to provide updates as we go along with the study, and at some point we'll get to a point. If you, from what Vertex announced publicly and what we've said as well, which is we hope to enroll the whole study this year, both studies, both sickle cell and thalassemia. We'll provide updates along the way, and we'll provide data cuts at various points, most likely at conferences. What's left to be shown, I think, to a large extent, we're very pleased that the pattern of fetal hemoglobin elevation is consistent across all the patients. And regardless of genotype in the case of thalassemia or the severity of the sickle cell, we obviously want more numbers just to confirm that, and also follow the patients for longer. The first patient treated in thalassemia is now out 2 years and is still cured, quote unquote. And I think we'll just get a richer data set with more numbers and more duration of follow-up that should give not just us, but all the treating physicians confidence that this therapy is in fact a one-time curative therapy that can have durable lifelong results.
Yeah. And then in your data set, there was, I think, only one patient with beta 0 genotype thalassemia. This is sort of a subset of patients where others had some more problems initially to generate high enough hemoglobin levels. I guess maybe talk about your confidence level that with CRISPR, with your CRISPR gene editing technology, that is something that's less of a concern for you.
Yeah, I think a couple of things give us confidence. One is, the first patient treated with this therapy in beta 0, while not technically a beta 0, was an IVSI 110 mutation, which acts like a beta 0 mutation. It's very low endogenous beta globin production, very high transfusion burden. And I think we saw a remarkable level of fetal hemoglobin production in that patient. The other thing we're seeing is, even some of the other mutations there, we see a pretty remarkable fast elevation of fetal hemoglobin to the levels required, and then there's an equilibrium at over 9-10 grams per deciliter. So I think, based on the data we've seen so far, we have confidence. But I think to your point, we just need to enroll and dose more beta 0 patients so that we can actually confirm that in clinical trials and see that goes up durably.
Okay, very good. And then, we already spoke about your regulatory interactions, so it sounds like that's forthcoming. And what sort of a time frame for potential update to the investment community on that front?
Yeah, we'll provide an update this year. As you know, these regulatory meetings, once you have a designation like RMAT or PRIME, you end up in a continuous mode of discussions. So it's not like one regulatory interaction where you have all the answers. So I think we're continuing to work with regulators on the CMC front, on the clinical protocol front, to make sure that we have full clarity before we update the street on what a path would be for filing. But we hope to do that this year.
Very good. And then just in terms of, labeling strategy and commercial strategy longer term, there obviously is a competitor in the space. How do you think about, balancing maybe speed to market versus, generating a very competitive label with clinical outcomes in order to drive reimbursement and a strong label that helps you to gain traction in the space?
Yeah. In the sickle space, which is the major market, there's not too many degrees of variable on the label. I think it's, we hope that this is a therapy that will be approved and applicable to anyone who has more than 2 VOCs per year, but that's some of the technicalities around it. But for severe sickle patients, I think there's not that many degrees of freedom. In thalassemia, I think there is a question of the different genotypes, but what we're hoping to do is do a pan genotype. I mean, this helps every transfusion dependent thalassemia patient regardless of genotype, and that's sort of what we want to show with our study. Now there's the question of pediatric and including pediatric in the label, but we could get to that at a later time. And so those are some of the choices around how long you want to wait before filing versus accruing data in populations that expand the label. Those are choices we'll make jointly with Vertex this year and update the street.
Okay, very good. And then in terms of manufacturing scale and CMC requirements, again, it's an area of difficulty for others. What do you have to accomplish here, in your opinion, in terms of either scale or potential commercial CMC requirements?
Right, and the scale up is a very different phenomenon in autologous cell therapies than it is with allogeneic or biologics. I think what you're doing in terms of scale is simply repeating unit operations many times and adding more suites and more operators to the mix. Now we do need to do a few things, which is, the priority for the regulators is to make sure that every product released meets the quality bar. There's not a high level of variability in the output from the manufacturing process. And so there's a set of things around release assays and potency assays, et cetera, that need to be fully buttoned up, obviously. And we started off that way, by the way, knowing that would be the case to make sure we have a lot of those things developed early on, and so that nothing catches us by surprise. And then the second thing is, as you scale up, I think you need to make sure the suites you're adding, the CMOs you're adding, and capacity you're adding is all qualified and comparable to what you started off with. So those are broadly the two challenges there, but we don't anticipate them being significant challenges at this point, and we're in the process of scaling up to meet the demand for hundreds of patients from the scale we have today, which is tens of patients per year.
Yeah. Okay, great. And then fast forwarding towards commercialization, I guess, what is your philosophy around pricing? Others in the field have made statements and proposed different pricing models for gene therapies. And how do you balance that with access, in particular in sickle cell, where there's a high proportion Medicaid population?
Yeah, I think these commercialization is going to be different from what we've known as an industry. We just hired our chief commercial officer 2 weeks ago, Philippe Drouet, who came to us from Merck and led the Keytruda franchise there. I think on coverage for sickle cell, it'd be a shame if this country cannot provide coverage for these patients suffering from these diseases. And we're not talking thousands and thousands of patients here. I think in the early going, it's hundreds of patients, and these are patients that are they're facing severe disease. They're like, they have to go to the hospitals all the time. There's a cost to the system from these patients. They're not productive citizens of society. And I think we'd be working with both the state level as well as the federal level to make sure that there is reimbursement for these patients and there's access. I think it's too early for us to comment on our own pricing. There are some benchmarks out there with other competitor drugs that have been launched already. We'd be very open to different pricing models, and whether it's an amortized model over time, or other ways of changing the pricing from one time to a more balanced pricing over time. But I think ultimately, we believe that the access will be there because for this patient population that suffered for so long, it'd be a shame if there's no support from the health system.
Very good. All right. So I did want to spend also some time to talk about your CAR T programs, maybe then shifting over to CTX 110.
Mm-hmm.
So we saw some very early data again from the phase 1 carbon study late last year in non-Hodgkin lymphoma. Maybe could you just update us how this study's been tracking towards identifying a potentially recommended phase 2 dose and preconditioning regimen, et cetera?
Yeah, I think we were quite encouraged for our first data release in oncology with allogeneic platform to see nearly 50% complete response rate out a month or so after treatment at the high doses was something that was to us very promising. I think except for the one patient where there was a patient death at high dose, and we now believe that death wasn't caused by the CAR T or ICANS. It was caused by viral reactivation. And we are actually probing higher dose levels. We're back to probing higher dose levels. We believe that we have something here that's an active drug, and we're seeing very good responses. Now, the key, as we all know, is durability. We want to make sure these responses are durable. And I think we haven't so far needed to make a lot of changes to the lymphodepletion regimen. But what we do want to follow is with the single dose, how much durability do we get? Okay, and if that durability is very long, we have a single dose as the expansion arm and potentially pivotal registration arm. But if we need to go to a redose schedule, maybe not for all patients, but for some patients, that's something we're going to pursue as well. And so there's a little bit more of the dose finding, a little bit more of the optimization here to identify the sort of the expansion dose or the pivotal dose or pivotal regimen, I should say. But we're getting there, and I think in spite of COVID, we're making good progress, and we'll hope to update this year at some point.
Okay, very good. And then maybe just briefly on 120, your BCMA CAR T product. We haven't seen any data yet, and maybe just update us on how that study's been going and tracking behind 110 maybe, and how investors should think about forthcoming updates as well.
Yeah, I think the thesis for 120, which is against BCMA, is the same as 110, CD19, which is we have a known target where CAR Ts are active. Let's prove the allo platform against these known targets. From a enrollment standpoint, the dose escalation standpoint, I think it's a very similar pattern as 110. For the course of the study, we have to go through different dose levels. We're starting to get to more meaningful dose levels now. I think our initial update will probably be like 110, a company update, followed by a more thorough update at a scientific conference. So that's progressing nicely, and so is our third program, CTX 130, which have 2 trials ongoing, one in heme malignancies, the other one in solid tumors.
Yeah, I'm really curious to learn about that. I think you did say you plan on potentially providing updates or clinical data maybe on the 130 program this year, which would be the first allogeneic solid tumor CAR T data in general, I think, to my knowledge. Maybe talk about how CTX 130 is designed to potentially succeed in solid tumors, which has had some difficulty historically, where you see the most compelling path forward.
Yeah, I think the key difference between 110, 120, and 130. So 130 has an additional edit that is designed to prevent exhaustion in these CAR Ts. Our hypothesis is the main reason CAR Ts haven't worked in solid tumors is because of exhaustion in the tumor microenvironment. And this edit helps overcome some of that exhaustion of these CAR Ts. So, we're obviously starting, as you know, we're starting our 130 trials in solid tumors in renal cell carcinoma, where there's a strong unmet need, notwithstanding some of the data we see recently with PD-1 inhibitors and in combination with TKIs. There's still an unmet need, I think, once you get past that first line of therapy. And we hope to start, if there's good data that we see in renal cell carcinoma, CD70 is also expressed on a number of other solid tumors. So we would expand into other solid tumor types soon after.
So how far in are you into the study, and can you incorporate any learnings from the other programs to accelerate that program?
Yeah, I think, I think all these trials, the major limitation in getting it going is the dose escalation period, because for regulators, you're mandated to go in a 3x3 design or 3x4 design with, serial dosing to get past a certain dose level. And that just takes time, especially during COVID environment. But we're making steady progress. We're not wasting any time or losing any time there. And once you get to certain, more meaningful, therapeutically meaningful doses, that's when really the trial starts. And that's where the art of dose finding, the art of optimizing lymphodepletion, all that comes in. And at that point, we'll again provide a company update, most likely followed by academic conference.
Okay, terrific. And then I know we're running up on time, but I did want to give you an opportunity to speak about your Regen Medicine program, and specifically around the type 1 diabetes product, and maybe just very briefly how that concept compares to maybe other regenerative medicine approaches that are being worked on for type 1 diabetes.
Yeah, I mean, the concept is very simple. I think it follows the Edmonton protocol, where it was realized that you can take cadaveric islet cells. Islet cells are the cells of the pancreas that produce insulin and respond to glucose in our bloodstream. And if you can inject these cadaveric islet cells into patients with severe type 1 diabetes who are immunocompromised or immunosuppressed, then they don't need a lot of supportive insulin. They can actually regulate glucose through these cells. And so the question was, can you just do that from stem cells? And the only issue is, yes, you can, as our partner Viasat have shown. The only issue is the cells don't last very long without immunosuppression. And that's where gene editing comes in. And we now make 5 edits in these cells to prevent elimination by the host immune system. And we hope that that product can actually be applicable not just for type 1 diabetes, but also for type 2 diabetes. So I think, we're just getting started here. I mean, we went from 1 edit for sickle cell and thalassemia to 3 edits for our immuno-oncology programs to 5 edits now for Regen Med. And, we have the ability now to go up to 10 edits in our programs. And so the more we learn about immunology and the immune system, the more we're going to tweak these cells and make the products better and better and better. We're already seeing promising data across a number, at least 2 of our programs. And we hope that we'll see that across all our programs. But I think cell therapy is here to stay, and I think it's going to be a major part of medicine.
Very good. So with that, I think that's a good concluding thought. I think it's time to wrap up. 4 p.m. Friday afternoon on the U.S. East Coast before a long holiday weekend. So I'd like to thank everybody for tuning in. Sam, thanks for your time. And we look forward to a very busy year for CRISPR Therapeutics this year.
Terrific. Thank you, Michael.
Thanks so much.